For this video, we are going to discuss lipids and their role in biology and what they look like, their structure. And as I've talked about a lot, their structure gives the lipids their physical properties and their function. So we're going to really, as a chemist, I'm really going to focus on their structures, and then we'll talk about their biological roles and how they're so important in biochemistry. So if we jump right in, we have to understand that when we say lipids, there they all are. Right? They're very diverse. There's many different structures to look at. It's not like when I said an amino acid. And when you look at an amino acid, people would draw this generic structure right here. Right. That generic structure is an amino acid. All amino acids are going to look like this, or should I say L amino acids would have that look, right? All you would have to do is change the side chain, boom. But here you can see that we have a species like this. So well, let's back up before I jump into that one. Okay, lipids have a component of a fatty acid. So this right here is a fatty acid. Fatty acids compose of a long carbon chain right there with a carboxylate head group right there. And that molecule right there, the fatty acid, there's a special name that describes its physical properties. You see that we have like a polar end, or a polar end here, and a nonpolar tail. Or we could say, hey, this is going to be this part right here. That's hydrophobic. So we have a hydrophobic tail. And why is it hydrophobic? Well, it's all carbon. So it's nonpolar. And hydrophobic species don't associate with water very well. They don't dissolve in water. It's kind of like taking oil, dropping it into water. The oil is just going to float. It's not miscible. And then this part right here, the polar head group right there, that would be what? Hydrophilic or water-loving. So we have a molecule that has its two physical properties. One end is hydrophobic, doesn't like water, but the other part of the molecule is hydrophilic and loves water. And we have a word that describes that relationship or that kind of structure here. And you will see the word amphiphilic. Amphiphilic describes that. We have a hydrophobic part and a hydrophilic part. No, hydrophilic part and a hydrophobic part. You could use another word that looks very similar to this, and it's called amphipathic. And I'm going to erase the amino acid here. Amphipathic and amphiphilic, same thing. Okay? It's describing this process that I've just gone over. But those two words are used interchangeably. They mean the same thing. Now we have this. So we have, now what we can do is take fatty acids and stick them onto molecules such as glycerol. So if we zoom in here, I can't zoom when I'm on the pen tool. Let's see here. Apparently not. Let me do it a second ago. Let me pause the video and try to figure this out. Well, I can't seem to figure out how to get it to zoom in, so my apologies. So on your personal slides, you're going to see this molecule glycerol. And we're going to look at other slides that zoom in on that. But the point is that we have this molecule glycerol, and then to that glycerol molecule, we can add fatty chains or fatty acids. And that's what you see right here in this molecule. And if you could see here, here's the fatty, let's see. There's the fatty acid right there. And here's a fatty acid right there. But you notice how those two fatty acids look different? This one right here has no double bonds, whereas this one does have double bonds. So you can change the look of the fatty acid. To this glycerol molecule, you can attach all the same fatty acids or different fatty acids. And when you attach fatty acids to a glycerol molecule, we get something called a triacylglycerol. So that would be called a triacylglycerol, like that. So that's another look that you're going to observe. Okay. And then right here, we have a sphingolipid. So now we're talking about lipids now. Okay. We have fatty acids, triacylglycerides, or triacylglycerol. And now we have, what do we see here? It looks a little bit different, right? But we see a fatty acids and something called a sphingosine. And then we're going to have different types of head groups. So this whole slide is to illustrate diversity when we're talking about lipids. Right here, that's a steroid. And then we have a different look here. We have ubiquinone right here. Now we're going to start delving into kind of like what are their biological roles. And then after we look at some of their biological roles, then we can talk about their structures and dissect the classifications and how they really look, okay? So you see on the slide here, what are lipids used for? Storage of energy, okay? Fatty acids, they have a lot of carbon that can be used to, it's fats, basically. They're fats, and we can process fats for fuel or process fats for energy. Okay. Insulation, they are fat, so, you know, like polar bears and seals and all that, they have a lot of insulation. I know some animals, they put substances on like their feathers, like for ducks example, to repel water, and it's because of the lipids that are in those molecules. We'll focus a lot on membrane structure. That's huge for this class. Cofactors for enzymes. We've talked about enzymes before. And so when you go back and review enzymes, you'll be like, hey, now you are like, we've talked about lipids before, and now you can make a connection. Okay, signaling molecules, that's huge. That will come in later chapters. And then pigments, which is kind of interesting that it gives the color to vegetables and to birds, and it's just interesting. And then it can also be with vitamins. So once again, very, very diverse in their purpose, not just only their structure. Okay, this is just a cool slide. Okay. I can really appreciate colorful birds. Growing up in a part of the country where colorful birds was not a thing, you had blackbirds and seagulls. It's kind of really the breadth of the birds that I saw growing up. And so moving to Florida and Pennsylvania, seeing a cardinal for the first time was like, it was an experience. But the color, yeah, you can see here, fatty acid chain, or not a fatty acid chain, but a hydrophobic chain of carbons. Okay. Okay, now let's get into the nitty-gritty details of the structures of lipids and how they're composed. And so we want to talk about the similarities and the differences. And so when you look at this slide right here, I'm going to expect you to understand this slide in detail. Okay? So for example, we have the glycerol phospholipids. They are under a class of phospholipids, and we'll talk about this all in a minute. But you can see what they're composed of. A glycerol phospholipid is composed of glycerol, 2 fatty acids, a phosphate, and an alcohol. So I want you guys to be able to basically memorize this slide, okay? And the function or the components of all these lipids. So let's first focus our attention on the storage lipids. You can see here the triacylglycerols right here. So these storage lipids that's one representation, right, of the triacylglycerols. If we come back a few slides here, we can see an example of one and how I talked about the fatty acid chains can be different. It all depends on the species. But so you see the structural component of it, okay? Now, why? Well, first off, I'm just going to state a fact. There is an advantage for storing these in our cells and in our bodies because lipids can be used as a fuel source. Why does the body want to store fats? There's an advantage to storing fats in comparison to storing carbohydrates. So let's talk about 2 advantages here of fats being stored as a fuel source. And so the first is that when you take a look at the fatty acids, there's a lot of carbons. And we have to take those carbons and oxidize them. And when we oxidize them, that's how we get the energy that we need. And so when you compare a fatty acid to let's say, glucose molecule or carbohydrates, the fatty acids are more reduced. And because they're more reduced, that means they have to undergo more oxidation to get converted into, well, it just requires more oxidation to break it down. And so every time the molecule gets oxidized, energy is released. And so when you compare a carbohydrate, it's more oxidized, so it's going to have less oxidizing reactions to get converted to what it needs to be. And so what fat molecules offer us is more than twice the amount of energy you're going to get in comparison to using carbohydrates as a fuel storage mechanism. Another advantage of storing fats for fuel is that because they're fats, they're very hydrophobic. They don't like water. In comparison, carbohydrates have a lot of oxygens in them, and so that attracts water. And so if you store a lot of carbohydrates as a fuel source, then you're also going to bring a lot of water with it. And that gets very, very heavy. And so fats don't have that problem. Less water is going to associate with it, less weight to have to carry around. Now, there is an advantage of having carbohydrates stored as fuel as well, and that is carbohydrates can be broken down into energy sources faster than fats. And so fats pack more bang for your buck. It gives you more energy, but it takes a little bit longer to get it into a usable form, whereas carbohydrates, quick. So you can see for different scenarios, like if you're getting chased by a bear, your cells and your body's going to be like, let's use the glucose or the carbohydrates. We need energy fast. Whereas fats would be just slowly releasing the energy that you need when you're not in fight or flight. Okay. So triacylglycerols right there, composed of a glycerol backbone and has the 3 fatty acids. And those 3 fatty acids can be all the same or different. Let's make sure we understand what glycerol looks like. Let's draw that right here, shall we? Like that. All right, let's double check it. Back to the slide right here. You can see, what does glycerol have? 1, 2, 3 carbons. 3 carbons, 3 alcohols. So when you take the glycerol and attach the 3 fatty acids, what type of linkage do you have between the glycerol molecule and the fatty acids? All right. What type of linkage do we have here? Let's just draw one of those linkages on this molecule right here. What's going to happen when you form that bond between the fatty acid, you are going to have so forth. The chain will continue to go on and on and on and on. Well, within limits. But what type of linkage did we form to the glycerol molecule with this fatty acid? What is that linkage right there? That's the linkage. That would be an ester. So we have ester linkages when we make the triacylglycerols. Now moving on to membrane lipids. That's really, really big. You can see under that umbrella of membrane lipids, okay, we have these 3 different types right here. And we're going to spend some time looking at their differences. But in a quick view of it, okay, we have something called phospholipids. What are phospholipids? Well, you can see that they just have a phosphate group. Okay? And then within the phospholipids, we have the glycerophospholipids. And why do you think it's called the glycerophospholipids? Because we have the glycerol molecule right there. But then we can replace that glycerol with something called the sphingosine. And now we have the sphingolipids. But under this class, both of these are still considered phospholipids because they both have the phosphate group. When you take a look at the glycolipids, glyco, what's that suggesting? Carbohydrates. Okay. So we have the glycolipids right there. We see right here, we have the glycerol. We have two fatty acids attached to it, but we call it a glycolipid because it has a sugar attached. But under the glycolipids, we can branch off again. Why? Because for this glycolipid, it has a glycerol component to it, whereas this one also has the carbohydrate, but it has a sphingolipid or a sphingosine. Now, what you need to understand here is that, well, let's just continue on and finish this class right here. We have archaeal ether lipids right here. Okay, and you can see they look a little bit different. They have two glycerol molecules. They have some phosphates there. Right. And they even some of them tack on another glycerol. So these lipids right here are going to be found in the membranes of species that are in extreme environments, like near at the bottom of the ocean where the volcanic ash, not ash, but the volcano is fuming out all the hot gases, and the liquid or the water is like extremely hot. But scientists go down there and they're like, there's things that are living right by those vents. And so those species need a membrane, a cell membrane that is so strong that it can withstand those extreme conditions. Like if we took our cells and brought them down to that pressure and that temperature, our cells would just blow apart. They wouldn't survive. And it has to do with the structure of those lipids. And you can see that they're different than these two classes right here. And these two classes right here, especially the phospholipids, you will find both of these in membranes of cells. Okay. Now so when I'm circling all these different components, you can see that they're color-coded. Okay, we have the peach color, the yellow, and the blue. And those are, we're going to give those colors some classifications. So everything in that peach color. So you see, well, I mean right here, all that stuff, okay? Everything in that peach color, we're going to classify that as the backbone. So we have the glycerol backbone of lipids, and we have the sphingosine backbone of lipids. Everything in blue on this graph right here, that's called the head group. And what you're going to see is that, let's say we look at the glycerophospholipids. Yeah, it has an alcohol, but this head group right here, shown in blue right there, that can change to give you more variety of the glycerophospholipids. depending on what type of group you have. So the head group can change, and we're going to see a lot more of that variety in later on. And then in yellow, you can see that's already written out. Those would be the fatty acids. Okay, so those are the three main components of a membrane lipid. Okay, we have the backbone, the head group, and the fatty acids. Whereas, you can see in this bullet point right here, there are some lipids that don't contain fatty acids. And those would be cholesterol and some vitamins and some pigments and so forth. What would be another one that we saw on this slide right here? Ubiquinone right there. Okay. Right there. Okay, is there anything else? So this is still kind of like an introduction of the main components, because we're going to actually start, instead of looking at rectangles and colored rectangles, we're going to actually start looking at the structure. So I'm just double checking. I think that looks good. Okay, now we're going to spend some time looking at chemical structures. So we are going to focus our attention now on the structure of the fatty acid and make sure that we understand some basic terms and basic nomenclature. And the first thing is that when we look at the fatty acids that we're going to look at, the majority of them have a length of 4 to 36 carbons. Okay. And interestingly, we observe that these fatty acids are always going to be even numbered of carbons. And then a lot of these fatty acids that we're going to look at, they're not branched. You can see this fatty acid chain is just straight. There's no branches of carbons coming off of it like that. No branches. We have those words right there: saturated, monosaturated, and polysaturated. Sorry, saturated, monounsaturated, and polyunsaturated. So let's break down what those terms mean. Okay, saturated simply means when you look at the fatty acid chain and you look at all the carbons, that chain is saturated because every carbon has the maximum amount of hydrogens that it can have. So that's an exercise for you. Go through, look at that molecule right there, look at all the carbons, and you'll see they have the maximum amount of hydrogens per carbon. That's what it means to have a saturated fatty acid. A monounsaturated is this guy right here. That has a double bond in it. And when you add a double bond, what is that going to do to the hydrogen count? It's going to reduce the hydrogens by 2. Okay? So if we compare this guy, how many carbons are in this fatty acid? I'm ignoring the carbonyl carbon. Let's just count them. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. Over here, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. So the difference between these two fatty acids is this one's going to have 2 less hydrogens. And because those 2 carbons right there, they don't have the maximum amount of hydrogens. They're missing some. So that's what makes it unsaturated. And then you can see with the mono, that's saying that there's 1 double bond. And then a polyunsaturated would be adding more of those double bonds. Now, what's interesting about this particular double bond, I'll ask you a question. Is it cis or trans? Coming back to our orgo knowledge here. That would be a cis bond right there. So that would be a cis fatty acid. We could also call it a cis mono unsaturated fatty acid. Right? You've probably heard of trans fats. Well, that would be fats that have trans bonds, and that is very bad for our health. Okay, naming fatty acids. Now, naming fatty acids, you are going to see lots and lots of tables of fatty acids. Okay? So when you look at this table here, we clearly have a structure, we have a systematic name, and then we also have a common name. Okay? You don't need to memorize these tables, but you should be able to answer a few questions and a few things about these fatty acids. These are some common ones that we're going to deal with. And so in this next slide right here, you see these numbers right there, 16, 1, delta 9. I want you to know what that means. I don't necessarily want you to understand, hey, this acid has that chemical formula. I think that's a waste of our time. But there's some parts of the nomenclature I do want you to know. And so that's what we're going to talk about right now. Okay? So the first thing here, we have a systematic name here. Let's take a look at this. Let's look at the components here. Okay, what does octa mean? That means 8. What does deca mean? 10. So when you look at this numbering scheme here, how many carbons do we have? We have 18 carbons, right? And it's really important. How do you number the fatty acid chain here? You start with the carboxylate end. The carbonyl carbon is always going to be carbon 1, and then you just number it. 1 through 18. Okay? So that's what this number right here, can you see it? You can barely see it, but 18 right here, that's telling us how many carbons we have in the fatty acid chain. And remember, that includes the carboxylate carbon. And now we have the 1. What does the 1 represent? It's telling us that we have 1 double bond. And what type of double bond? Well, it tells you right here in the name. It's a cis double bond. Okay. And then the delta symbol, the delta 9, what does the delta 9 tell us? Where that double bond is. And you can see counting 1, you bump into the double bond at carbon 9. So that's what that means. So if I give you just that number, those numbers right there, you would be able to draw this molecule. Would you be able to name it this whole thing right here? I'm not too worried about it. Okay. Or what is its common name? Does this one have a common name? Let's see. (18.1) right there. That was the. So that's the systematic name that we just found. Oleic acid would be the common name. So I am not concerned that you could look at that structure and say, hey, that's oleic acid. Oh, right there on the slide. Okay. I really want you to be able to look at that. Okay? Now we have another way, another scheme of numbers. Right there. Omega. Now the way these two numbers, the 18 and the 1, that's the same compared down here. But what's the difference between this one and this one? Well, here we have a delta symbol. Here we have the omega. And the omega is a different way to telling us where is that double bond. Have you guys heard of omega-3, omega-6 fatty acids? That's been pretty big in the last 10, 20 years about, oh, these are some fatty acids that we need to consume in our diet. They're very, very healthy for us, right? So this particular fatty acid, it is a omega-9 fatty acid, and that's because we count now from the opposite end. 1, 2, 3, 4, 5, 6, 7, 8, 9. We bump into the double bond, 9 carbons in, from the end. The numbering scheme is now just different with that omega. So if we take a look, here's the tables. We already looked at this one. All right. But here, let's look at this. This one, that number gets a whole lot more complicated. Look how many numbers are by the delta. So the 20 tells us we have 20 carbons long. The 5 tells us that we have 5 double bonds. And then the delta tells us where those double bonds are. There's the common name right there. But if I look at this molecule, if I draw this molecule and I ask you, what type of fatty acid is it? Is it an omega-1, 2, 3, and so forth? And what you're going to do is start from over here, okay? And look, where do we bump into the first double bond of the 5? We bump into it at carbon 3. So when there's multiple double bonds in the fatty acid, and we are talking about omega, okay, omega fatty acids, and we want to classify this fatty acid, it would be classified as a omega-3 fatty acid. It doesn't matter that there's multiple double bonds. That's just a big blanket term, omega-3 fatty acid right there, because we bump into the first double bond at carbon 3. But remember, that's counting from the end to the carboxylic or the carboxylate end. And they're very important for our health because we need them, but our bodies can't make them. So we have to get them through our diet. Okay? So where are some omega-3s found? Avocados, oils, olive oil, I believe. Okay. Let's see if there's any other nomenclature part that we need to talk about. Now let's take a look at some physical properties of these fatty acids. And I'm going to talk about the chemistry behind why we have these different physical properties. So when we take a look at solubility, I hope it makes sense that longer the chain of the fatty acid, less soluble it's going to be. Because longer the chain, you're increasing more carbon which is more grease, more hydrophobic character. And so the longer the carbon chain, harder it's going to be able to be soluble in water. Because water is polar, the fatty acid chain is nonpolar. And we've learned this term in chemistry, like dissolves like. And so water is not like a long fatty chain because water is polar and the chain is hydrophobic or nonpolar, so they don't mix. Melting point. You can see that the melting point is going to decrease as the chain length decreases. Excuse me. Why is that? Well, we just think about it. I think about it just with surface area. And I'm going to use my arms here. If I have short arms and I bring the two fatty acids, both of my arms, and put them on top of each other, that's the van der Waals forces, right? And molecules are going to pack together. When there's more surface area, meaning a longer fatty acid, there's going to be more surface area for those van der Waals interactions to occur. And more van der Waals forces will increase the melting temperature. Now we have that other point right there, that the melting point decreases as the number of double bonds increases. So that's saying more double bonds is going to make the melting temperature go down. What's the chemical explanation for that? Well, in this graphic, you can see that we have a saturated fatty acid here and an unsaturated fatty acid there. What does the unsaturated fatty acid have? Clearly a double bond in it. What does that double bond do? It introduces a kink. Right? Now come back to surface area when two fatty acids come together to interact. How do you think two fatty acids that are saturated are going to interact compared to two fatty acids that are unsaturated? How's that packing going to look? Well, that's what you see right here. The saturated fatty acids are going to pack together very nicely. And because they're packed so nicely, you see all those van der Waals forces in between there, right? And all those van der Waals forces are going to increase the melting temperature. Whereas when you look at a unsaturated fatty acid, you can see that there's still going to be van der Waals forces, but there's going to be less than the saturated. And that's why a unsaturated fatty acid is going to have a lower melting temperature. So if we come back here, and look at this slide right here, this is kind of interesting. So we know olive oil is liquid at room temperature, whereas butter is solid at room temperature but soft. And then if you have beef lard or animal fat, it's harder at room temperature. They're all lipids, they're all fatty acids. But why is one liquid and one solid? It has different physical properties, obviously. And what contributes to these different physical properties? It's the composition of the fatty acids. Olive oil, butter, and beef fat all have fatty acids, but their fatty acids are different. So in beef, we are going to have a lot of unsaturation. Okay? Sorry, I misspoke. Beef is a saturated fat. Lots of saturation. So what does the beef look like when it looks at its fatty acid chain? It's going to have just fatty acid chains that look like this. right? Whereas the liquid, or sorry, the olive oil, which is a liquid at room temperature, what is its fatty acids going to look like? Oh, that's trans. We got to go this way. And it erased it all? Oops. So instead of me trying to draw, let's just go back here then, shall we? So this is going to be olive oil, and this is going to be beef fat. Beef fat is saturated fat, no double bonds, more van der Waals forces, higher melting temperature. Hence, it is solid at room temperatures. In contrast, when you look at olive oil, because it's unsaturated, it's going to be liquid at room temperature. I think that's pretty cool. So depending on, when you look at this slide a little closely, you can see the size, and then saturated versus unsaturated. So it's a balancing act. How many double bonds, how many carbons, all of this influences the melting temperatures of these fatty acids. So let's highlight these differences when we take a look at the tables. So when you look at this table right here, let's focus on the melting point. You can see we go from 42 degrees and it's just increasing from 42 to 86. Why is there an increase of melting temperature? Well, it's attributed to the length of the carbons. You see that? Longer the carbon, increasing the melting temp. But now when we start introducing double bonds, things get a little bit more not so straightforward like in this slide, but there's still some information that we can get from it. So in the next slide, we're going to have to actually go back and forth now between the slides to make some comparisons. So in this slide, we saw the length. As the length changed, the temperature changed. Now let's compare, I think I want to compare 16 carbons has a melting temp of 63, but let's introduce one double bond. What's going to happen? Okay, before we go to the next slide, what do you think is going to happen to the melting temperature when we introduce a double bond? Is it going to be higher or lower than 63 degrees? Okay, let's go answer that question. 16 carbons, we add one double bond, what did it do? It went to 1 degree. 62 degrees difference. That's significant. Adding just that one double bond. And you can see that all the way through pretty much. 18 carbons, 69 degrees. You add a double bond, goes to 13. But notice, when you compare 16 with one double bond and 18 with one double bond, look at their melting temps. Why did they increase from 16 to 18? More carbons, longer chain. Right? So even with double bonds, longer the carbon chain, the better the higher the melting temperature is going to be. Now you can see here, when you compare these two numbers, why did we go down in temperature? Why did we go from 13 all the way to 1 to 5? Because we have added another double bond. More double bonds decreases the melting temp. Now, what do you think is going to happen when you go from 18, 1 double bond, 18, 2 double bonds, 18 to 3 double bonds? What do you anticipate is going to happen? The temperature go up to 211 degrees? That should make you do a head scratch. And that's exactly what I did. I'm like, wait. That doesn't make any sense. And it doesn't, because that is a typo. Nope. The actual temperature is a negative 11 degrees. Okay? Now, the harder comparison that you'd make is like, what if you compared 18 with 3 compared to 20 with 4? Which one's going to have the higher melting point? You could argue, hey, this one has 20 carbons compared to 18, so that should have a higher melting point, but it also has one additional double bond. So that, you're not going to be able to just think through that logically to predict which one's going to have the higher melting temperature. That you would have to have the data for. But up to this, before to this point, everything that I've talked about, those guiding principles, if you understand those, you would be able to predict melting temperature of fatty acids when you see them on exams or quizzes or anything like that. Talking about trans fats, because in those tables that we just went over, they were just comparing saturated to unsaturated with cis bonds. But what about double bonds? Okay. Well, thinking about it logically, between a fatty acid that has 18 carbons, that's trans, versus, let me write it down, okay? Carbon that's 18, that's trans, versus a fatty acid that has 18 that is cis. Between those two, which one do you think would have the higher melting point? Well, the trans would have a higher melting point than the cis. That's because in the trans double bond, there's no kink. This would be saturated. This would be unsaturated trans. And this would be unsaturated cis. Right? Now, I didn't draw the full fatty acid, but you get hopefully you see the point. That the trans kind of looks like the saturated. So the trans is going to pack together better than the cis. So the trans is going to have a higher melting point than the cis. But then you might ask, well, what about the trans versus the saturated? Which one's going to have the higher melting point? The saturated one is going to have the higher melting point. So if I show you an example here, if we look at stearic acid, let's see, I'll write it here. So C18 stearic acid, it has a melting point of 69.6 degrees Celsius. When you take and do a trans, 18.1 trans, that has a melting point of 44.8. And then if you look at 18.1 cis, that has a melting point of 13.2 degrees C. So there's some data to help you remember which one's going to have the higher melting point. So there are a few other ideas that I want to address, and then we will conclude this video. So one thing about the triglycerols, remember what the triglycerol looks like, right? It is composed of the glycerol molecule with fatty acids attached to it, okay? Now, some tidbits of information that you need to understand here. There's some like distinctions right there. Fats are solid and oils are liquids. I think that makes some sense right there. Okay, so I'm not going to read this slide to you. This is just, tidbits of information that you need to know. But this one right here, I want to talk about this point a little bit right there. Okay. When we take a look at glycerol, you see all those alcohols right there. Okay. That can hydrogen bond with water. So glycerol is going to be soluble in water. But when you take glycerol, glycerol soluble in water. Let's take a fatty acid. Okay. Is there just an undo button with this thing? I don't know. When we look at the carboxylate, so we have the amphipathic molecule there. So we could, let's just say, somewhat soluble in water, okay? Because it has that hydrophilic part right there. But so soluble, kind of soluble. But then when you take those two pieces and put them together to make the triacylglycerol right here, this guy is not soluble in water. Okay. It's not soluble. And that makes sense from experience, because when you look at fats and you actually have it in hand, you're like, if you're thinking chemically, you're like, hey, glycerol soluble, fatty acids somewhat soluble. But now I have this oil or fat and I put it in the water and it's not dissolving. What's going on here? Well, it has to do with the fact that you are getting rid. You're forming a ester bond. And that ester bond kind of gets rid of its ability to interact with water as well as it could before it gets converted into an ester linkage. And I believe that's exactly what that says right there. Yeah, the so it's right there in writing, and I've just explained it graphically for you. Okay, so triacylglycerols, not soluble in water. So if it's not soluble in water, what words could we describe this? Triacylglycerols are nonpolar. All right. Fatty acid here was polar, but then when you put it on here, now the whole thing is nonpolar. Go to the next slide here. This is the tidbit of information that I've already talked about at the beginning of the video. So there it is in written form. to help remind you to remember and study that. And that's just a picture of some fat cells right there. Another interesting thing when we take a look at fatty acids, that we can take a fatty acid, palmitic acid, attach it to this other long carbon chain right here, but via ester linkage. With that ester linkage right there, what's going to happen is we make a wax. Okay? There's many purposes for nature to make waxes that are all listed right there. Okay? But one thing that I can relate to, I grew up around bees. We had a beehive where we harvested the honey. And so, yeah, bees make the wax for their honeycombs, and it's because, and the structure of that wax comes from molecules that look like this. So that's that one right there. Not much, it doesn't have much for biochemistry terms per se. I think it's just an interesting factoid. What is wax made out of? It's made out of the fatty acids. Okay, but it tacking on a big chain on the other side of the ester. It's kind of cool. Okay. Let's see. Okay, so this slide now, we've seen it before with the rectangles and all that, but now we are going to get into the chemical structures of all those components. But I think this is a good place to stop, and we will pick up the chemical structures later.